The growth hormone, which is also called somatotropin or human growth hormone, is produced by the anterior lobe of the pituitary gland in the brain. The pituitary gland is a small pea-sized gland that plays a huge role in regulating vital body functions and our well-being. It stimulates the growth of all tissues of the body, including bone. It is especially vital for normal physical growth in children. Its levels increase progressively during childhood and peaks during the growth spurt that occurs during puberty.
This is the coding sequence for our protein, which has 24 base pairs. The nucleotides which make DNA are adenine, thymine, cytosine, and guanine. There are two strands, coding and non-coding. To show the process of transcription, we will start with the 3 to 5 strand, which is the non-coding strand. The information in a strand of DNA, which stands for deoxyribonucleic acid, which contains sugar, is copied into a new molecule of RNA. Then we will take a single strand of RNA to match the non-coding strand of DNA. RNA polymerase, which is an enzyme, begins the process by opening the double helix so the non-coding strand can be copied and help assemble the complementary mRNA bases one at a time.
Ribosomes carry out translation tRNA to assist the ribosome by transferring over the amino acids needed. The molecule being translated is grouped into sets of 3 bases. These groups are called codons. Through the process of translation, each of these groupings get translated specifically into amino acids. We will now talk about the codon chart. Basically, it is a tool that helps us match the different amino acids to the different base pairs of the non-coding strands. For example, let's start with the first 3 base pairs. We take the first 3 base pairs to know how the ribosome knows what amino acid to give. So we have A, U, G, and C. Since we have our first 3, which is A, then we go to U, and then we go to the last one, which is G, which will end up giving us the amino acid, which is methionine. Due to tRNA having an anticodon that is complementary to the codon on mRNA strand, tRNA can then bring the correct amino acids determined by the codon. As the mRNA continues into the ribosome and gets towards the end of the first 3 sets, the amino acid goes on top of the other amino acid to the right of it, for this is the protein being built as it continues. The reason it piles up is because the amino acids are the building blocks of proteins, so it will continue on until the very end of the mRNA. In conclusion, we have learned that the growth hormone is made using the process called protein synthesis, in which a strand of DNA is used to make RNA, which is called transcription. The mRNA makes its way out of the nucleus and captures the ribosome. Then it makes tRNA from each codon of the mRNA and makes the protein from the amino acids, which is called translation. Through the whole process of it all, we have learned so much. We have learned exactly how the ribosome takes every 3 bases to receive an amino acid, how the mRNA corresponds with a non-coding strand in order to be taken into a second process, which is translation, how each codon helps to make a whole long strand of DNA, and so much more. We appreciate the experience and hope to apply it in the future and to our future knowledge.